Greetings, everyone. Welcome to your third session for tonight. We're moving away from the cars and moving towards the science. More specifically, of course, we're getting towards enzymes. So how are we all doing tonight? How's your night been so far? It was a great class with Ed. It was good to see him teach. It's been a while since I've seen him. I guess in person you could say cameras count for being in person as well. Good to hear, everybody. Yeah, seriously, Stefan, it's snowing all over, but nowhere nearly as intensely as Boston. At least that's what the news says. I'm not in Boston. I'm in Colorado, where we want the snow. So that's a big difference between the two places. Good to see you, Sam. What's going on? Thanks, I appreciate that. We're going to have some fun today. We're going to get a lot into well, of course we said the science side of things. Specifically, we're going to get into enzymes for the next 55 minutes. Specifically, we'll be talking about the structure and function of different types of enzymes. We'll get into a little bit of nomenclature. We'll talk about the main categories and classes and give a couple examples of them. After that, we'll give you a couple graphs. Specifically, we'll stick with Lineweaver-Burke a lot today. We'll give you a graph and tell you how we could basically determine the properties. Specifically, we'll spend a lot of time talking about graphs in conjunction with inhibitors. So we'll talk about the four types of inhibitors. And then, of course, we'll talk about their effects on Vmax and Km and all that stuff. We'll even derive what Km even means in Vmax and all that kind of stuff. So it should be pretty fun. All right, with that stated, the first thing, as you know, we're going to talk about is the structure and functions of different types of enzymes. So let's get right into it. All right, everyone, big picture first. In fact, this is probably the highest yield of everything we're going to talk about today. Heads up. But what are enzymes? Let's get big, broad category here. Exactly, they are protein catalysts. They don't technically all have to be proteins. At this point in evolution, they pretty much all are. But exactly, they are catalysts. They make stuff go faster, and yes, pretty much they're biological catalysts, mainly proteins. You guys got it right, at least at this point in our evolutionary history. But exactly true, they make things go quicker. So let's talk a little bit more about catalysts. So what do catalysts do? What do catalysts do? Bingo, Mary's got it. We lower the energy activation. There's lots of things we could say here, which speeds up the reaction, exactly. So let's talk a little bit more about this lowering energy of activation. Lots of folks are saying it. How do we do that? Like, let's get technical. How would you lower the energy of activation in a very specific sense? Like, you don't have to talk about specific enzymes at all, but like, what are you actually doing here? What are you creating? I like that you're going to lowering exactly creating a favorable environment. Devon, so close. Let's swap out one word. What's the one word we want to change? Go with what Devon's saying right there. There we go, Maria's got it. Instead of intermediate, you would call it the transition state. So remember, the big difference is an intermediate is something that's isolatable. Like, for example, in an SN1 reaction, your intermediate is the carbocation, right? Here, we're not talking intermediates because there's no little hump in the curve right here. Instead, we're talking about the top of the curve, and as you know, the top of the curve is known as a transition state and carries the symbol that looks just like this, which would also make a pretty good sciencey, nerdy tattoo if you're into that kind of thing. Exactly, that's the transition state at the very top, and so it stabilizes the transition state, thus lowering the energy of activation, which speeds up the reaction. And Mary's got the good thing. Basically, it's putting the substrates and the different things we're sticking together closer to each other. So it makes it easier for the reaction to actually proceed. It's exactly true. So notice here, of course, is the original energy of activation or activation energy. And of course, with the enzyme, it drops it. So now, wait a second. What isn't changing? What is not changing? And Rose is getting a good word there. We'll talk about that in a second. I'm glad you brought that word up. You don't change equilibrium. You don't change delta G. You don't change the equilibrium of a reaction. And so notice, of course, you are lowering the energy activation, but there's no change in overall delta G. So there we go. We're going to combine what Ashley and Rosa are saying here. This is probably the highest yield of everything we talk about today. You have this strong divide between two big things. We've got the thermodynamic side of things, which talks about delta G. It talks about, well, actually, if you're talking about delta G, what are we usually referencing? So delta G is used to describe a certain part of a reaction. What part of the reaction do we use it to describe? Almost exclusively, to be honest. You got it. Spontaneity, totally true. We determine whether it's spontaneous or non-spontaneous. That's what we use Gibbs free energy for. So we're not changing spontaneity. We're not changing equilibrium. We're not changing delta G. All we're changing is the kinetics, and that's the word Rosa brought up. So we're changing the speed. We're changing the rate. We're changing the kinetics. So then, with that stated, are enzymes speeding up the forward reaction more or the reverse reaction more? It's both. It's a trick question, exactly. Because notice the forward reaction, of course, is much easier here. It'd be going down that hump right there. But the reverse reaction has also gotten a lot easier because it used to be this difficult. So notice any change in energy of activation, you're going to have the same change whether it's a forward or reverse reaction. That's why it is actually both equally totally true, Stefan. Nice work. And everybody else, we have lots of folks saying it there. Perfect. All right, so that's probably the biggest, highest yield, major point that connects this topic to all the other science topics that we discussed. Let's get a little bit more specific, though. So there's one other thing that needs to be true for something to truly be a catalyst. So we're not changing equilibrium, not changing thermodynamics. We're only changing kinetics. We're speeding up your reaction. And exactly, it cannot be consumed. It doesn't get used up. It can be regenerated. Enzymes aren't regenerated, right? But exactly as Michael says, a lot of other catalysts can be regenerated, like protons are good examples of catalysts. But the thing is, it enters and it leaves. It's unscathed. It's not consumed. It's unaffected, exactly. Or it can be regenerated. All that does mean the same thing. Good call. All right, so that's the first step. That's the biggest picture. That's more than anything else. After this, we're going to get more and more and more specific throughout this session. So next thing, let's talk about the two different models, namely the lock and key model first. So lock and key model is exactly what it sounds like. Basically, what's the yellow, the enzyme or the substrate? In this, what would we suspect given the sizes of enzymes? Generally, it's the enzyme, right? Enzymes are pretty darn big, right? They're proteins. They're big macromolecules, right? And so the enzyme forms around whatever the substrate is that we want to form, and it fits perfectly. So as you can see, that would be an example of the lock and key model. Now, is this exactly true, given our current knowledge? Not totally, not really, Sean's a good way to say it. It's a combination of two different things. Exactly, as William says, because whenever you put two things close together, they actually cause conformational changes in each other. It's true, it does look like that. I like it. It's an upside down little green hand holding a baseball. But it's a combination of two different theories. It's lock and model, or lock and key model, and what's the other theory name where in reality it's actually a combination of the two? Induced fit. There we go, everybody, you got it. So it's a combination of both lock and key model, which says there's no conformational change, and the induced fit model, which says as the substrate gets closer to the enzyme, it causes a conformational change in that enzyme. Basically, it folds around, and it actually grabs on and induces the fit, making a perfect fit there. And so as Debbie says, in reality, we know there's an interplay, but you do want to know the two different names of the models because you might get a passage on test day that does discuss these different theories of enzyme binding, and you want to have the general idea of what both of them mean. Big take home, though, as Debbie says, combination of the two. All right, so it's a both factor of the substrate changing the enzyme as well as being a perfect fit in the first place. All right, so before we get too deep into this, let's talk about the two main sites of an enzyme. We've got, actually, what are the names of the two different sites? Actually, I take that back. I apologize. It's labeled on the screen. You can say it if you want. I try to avoid asking questions where the answer's on the screen, but yeah, you guys are right. We've got the active site as well as the allosteric site. I apologize for asking a question whenever it's right there. I'll try to avoid doing so whenever possible. Yeah, so we got the active site right there. We've got the allosteric site right there. And so notice the active site is where the substrate, as you can see, binds. So in this reaction, we've got a substrate. When it binds the active site, that enzyme's catalyzing a reaction that actually splits it into two different chunks. This is a very generic version of some kind of reaction. All right, so that's the active site right there. Notice it's a pretty good fit for the substrate. The allosteric site, on the other hand, does what? So why do we have that other site right there? You can see it. I like it. It's non-competitive, we could say. It regulates. It binds the ligands and other types of inhibitors or activators, we could say. It changes the conformation of the active site, as Ashley says. That's exactly right. So, for example, here, if we want to talk about the allosteric site, notice the allosteric site can either activate or inhibit. It can do both. It really depends on what enzyme we're talking about. But notice that in this case, we have an inhibitor. When the inhibitor binds the allosteric site, notice how initially it had that nice perfect wedge so the substrate could fit in. In this case, once that inhibitor binds to the allosteric site, it causes that wedge to change shape. Thus, the substrate no longer fits in the active site. So, given what we know in our background knowledge, what type of inhibitor would this be? Since it does label it as an inhibitor. It'd be an example of a non-competitive inhibitor. Notice if the inhibitor itself is going to bind to your allosteric site and cause a conformational change in the active site, That's known as a non-competitive inhibitor. We'll go through these very well in depth later on. On the other hand, where do competitive inhibitors bind? And we'll get real specific with this soon, but I want to kind of do a little preview because I know you guys know this stuff. You got the active site, right? So an example of a competitive inhibitor would be if we had something else that had that same spiky shape like our substrate there, but was a different molecule. Maybe it has like a box on the top. And so if you grab that, stuck that into the active site, it would stay there, and it wouldn't be able to have, I guess you'd say the substrate wouldn't be able to bind in the first place. We'll get real specific with that later on today. We'll show exactly what that means and how it affects enzyme kinetics, most importantly. All right, so since we talked about inhibitors, let's go the other route and talk about coenzymes and cofactors. So cofactors, how about this? What's the difference between a cofactor and a coenzyme? What makes a cofactor a cofactor? I like it. Ashley's got a key word there. When we say inorganic, what does that mean? Because it doesn't mean that it's covered in pesticides, in GMO, and stuff like that. What does it mean when it's inorganic? It means that it is non-carbon, Lauren. That's exactly. Specifically, it means that it's not carbon-based. It could be metals. It could be other stuff too, but primarily, well, pretty much most of them are metals. But you got it. It means that it's not carbon-based. We, of course, are carbon-based life forms. We are organic. All things made of carbon, you can call them organic, regardless of the pesticide usage on them. So these are inorganic compounds. They are, of course, non-carbon. So pretty much they're all metals, right? So zinc, copper, iron, stuff like that. Things that are not, of course, organic compounds. Coenzymes, on the other hand, are just the other one. There we go, perfect. I don't know too much about P90X, but that does make sense, Harpreet. These are organic, right? Vitamins are good examples. They are organic compounds, like vitamin C, right, is a good example. We actually see that once in a while. It's a type of carboxylic acid on test day as well. All right, so vitamins, across the board, things made of carbon. Which, of course, would these be bigger or smaller than the metals on average? What would we expect? Yeah, bigger, because it takes a bunch of different atoms to make something organic. You don't just have a single carbon atom, and so generally it takes several of these put together, so they're pretty bit bigger, we could say as well, just because we have a lot more atoms going in. All right, so here would be a little example. Just to label this, that's your substrate. The green thing there is your enzyme, and that red dot would be your cofactor or coenzyme. In this case, I would guess it's a cofactor because it looks small and circular, so it's probably it's a metal, some kind of metal, which would make sense. And so basically what the cofactor does is it allows the interaction between the substrate and enzyme to go well. And so it helps, of course, the binding between the two. It makes it work. And so there's loads of different types of cofactors that we'll see. Zinc, copper, iron, a couple good examples. Vitamins, another good one. You don't need to memorize a bunch of different cofactors for the MCAT. All you need to know is what they are, what they do, and of course the difference between cofactors and coenzymes. So notice this is just helping catalyze the conversion of breaking that disk in half, whatever that thing might be. All right, so these, of course, would be the positives, right? Helping a reaction take place, the opposite of inhibitors that we'll talk about for the very last part of class today. All right, so let's get, as I said, we're going to get more specific as we go through this class. Every slide, we basically get deeper and deeper. This is just going to be a big list of the different classes of enzymes. There's a bunch of different enzymes that all fit into these different classes. We'll just kind of give the big picture and different examples of them. Some, Roberto, it's a mix. It's totally messy. Some do straight require it, though. It's true. Some do require it, but of course it's very messy. I don't get too much into the specifics. Pretty much everything is true. There's so many different types of enzymes that pretty much you could think of a good example for all cases across the board. But I would say both. It's a short answer. Exactly. That's all happening at once, all at the same time in microseconds, in our body nonstop. But as Debbie says, we don't need to know the specifics. You do not need to stress about it for the MCAT, but you are very likely to see a passage on enzymes on test day, where, of course, we'll get deeper into this. All right, so I'll just give you guys the list first. These is the class of the 5. You might notice that there's also a little mnemonic here that we can do. Notice enzymes are just a little hot, right? Because they're pretty sweet, they're pretty hot, they make stuff happen quickly. Just like, how you call fast cars hot? You guys know what I'm talking about, right? That car's, no, I'm gonna stop. But as Debbie says, of course, you could remember this for the general category of types of enzymes. All right, so with that stated, Debbie brought up a good point that I didn't mention on the previous page. Thank you for mentioning it, Debbie. What's the difference between a holoenzyme and an apoenzyme? Thank you for bringing that up. I'm going to mention this real quick before we go along here. Yeah, which is which? Which is which? See, Debbie knows what to say. This is perfect. So holoenzymes mean they do have the coenzyme or cofactor. Apo enzymes are without, exactly. So halo, complete or whole, halo, whole, similar word root, apo, without, exactly. So apo doesn't have the cofactor, perfect. Do be aware of those terms as well. Perfect. All right, so let's go through, of course, these 5 different enzymes. I like it. All right, so there we go. So a little hot, as you said. So what's the function of a ligase? As Debbie says, you can use word roots to figure out exactly what those mean, too. That's the cool thing. I like it. Put things together. Where do we see ligase a lot? We see, I know it's a class, but there is one we just say ligase in DNA, exactly. So, of course, when we're talking about DNA replication, where does ligase really come in? We're going to talk about DNA replication. I like it, Ashley. Bringing in the keyword, the big guns, the Okazaki fragments. It's in the lagging strand, as Maria says. So remember, the lagging strand, of course, we form these Okazaki fragments because, wait, which way do we make DNA? Which way do we make DNA? You got, we always make everything 5 to 3. We pretty much consistently talk about 5 to 3. I agree the reading frame is 3 to 5. But of course, we're making stuff 5 to 3 across the board. And so ligase is, of course, dealing with the strand that doesn't go 5 to 3. It's dealing with the strand that goes 3 to 5. And since we can only make DNA in the 5' to 3' direction, you got to start at the end of the sentence and work your way towards the beginning. And so once you make all these little chunks, you then have to glue them together with your ligase. That's probably the most common example of that we'll see. So as we said, it joins compounds together. Generally, it requires ATP to do so. Same thing's true when we talk about DNA ligase. That's a classic example would be, of course, ligase. Now, one other thing, though, that I do want to mention, we don't get too much into this today. It's more of a metabolism talk, and we do talk about this in your metabolism channel classes. What's the difference between synthetase and synthase? So synthetase versus synthase. What's the difference here? Bless. Perfect. So synthetase needs, or which one needs ATP? Bless brings it up. Which one needs ATP? Or energy in general, we would just say, to be more specific. And the naming's kind of getting a little wonky recently. So generally speaking, synthetases require ATP, where synthases do not require ATP. Perfect. So do be aware of that. We don't talk about synthases today. We do talk about it quite a lot in your metabolism channel class, though. So, of course, if you have not yet attended that, I highly encourage watching it. I taught it a couple weeks ago, so if you want to check it out in the archives, it's still there for you to watch as well. All right, so that's synthetases, ligases, we already talked about, 2 classic examples of this. Next, we have isomerase. So what do isomerases do? Debbie's got a good mnemonic for you once again. Moving back to, of course, glycolysis, you got it. It's basically rearranging the conformation. Totally true. So this would be, if we want to jump back to glycolysis, glucose 6-phosphate turning into fructose 6-phosphate. That would be an example of an isomerase that does the conformation. Additionally, another type of isomerase is known as mutase. Mutases just move one functional group from one carbon to the next. So like, the 3-phosphoglycerate moving it to 2-phosphoglycerate, just moving the phosphate group from one carbon down the line. So just it's isomerizes, same idea, just rearranging compounds. Exactly true. So isomerase, mutase would be another one to put in there as well. So isomerase or mutase, types of isomerases. They change the conformation. Perfect. How about lyase? What about lyase? You got it. So notice ligase brings things together. So ligase with lyse. Exactly, Rosie, you got it. No, not using water here. There we go. And actually, Stefan, you're totally right. It does not use water to chop. So it breaks compounds apart. We got a couple of different examples of these, like lyase, ends in ase, pretty common. The ones that do use water, though, to break things apart, those are what? So these don't use water. That's the next down the line, as Ashley and people are saying. That's your hydrolase. Perfect. Nice inferring the next step down the line. So hydrolases, of course, break compounds apart using water. Lyases don't use water to break stuff apart. So same idea, though. Main thing is they break stuff apart. So peptidases, nucleases, lipases, we got a bunch of different examples. Of course, all of these will do so. How about oxidoreductase? We can use our word roots here. Perfect. Ashley's got the root, right? So ox and red in the same thing. We're talking about transferring electrons. Let's talk specific because this is a super high yield idea. So once again, is oxidation or reduction, which one is gaining, which one's losing? Yeah, Emma knows the mnemonic. Oil rig is one example. Oxidation is loss of electrons. Reduction is gain of electrons. Or if you're a fan of lions, some people love this mnemonic. I like oil rig. I don't know why I like oil rig. Maybe it's a, mountain thing. But Leo goes gur. You guys all ever talk about that one? So loss of electrons is oxidation. Gain of electrons is reduction. There we go. Mary's got it for you in the chat. You got to know which is which. But one thing I do want you to be aware of, there is no such thing as oxidation without reduction. Because if you're going to remove electrons in oxidation, they have to go somewhere, so you have to be reducing something. You can actually use that a lot on test day to your advantage, because if you identify one reaction as a type of reduction, you know there's got to be an oxidation there as well. So be aware, there is not oxidation without reduction. They come in pairs. That's why we call them redox reactions to combine the words together. So, of course, oxidoreductases will transfer the electrons. So, of course, you're going to be both oxidizing and reducing. Cool. Well, it depends what you're talking about, Harpreet, but cations have already been oxidized, you could say. So, cations have been oxidized. So, then if it's already a cation, well, I won't get into the agents. We have a whole session on that. I won't get to it. But you got it. But cations have been oxidized, you could say. That would work. Remember, there's also different oxidation states of cations. So, for example, you could have iron 2+ or iron 3+. So, in going from iron 3+ to iron 2+, what happened to iron? So, if iron went from iron 3+ to iron 2+, they're both cations, right? But it was actually reduced once it turned into iron 2+, because you became less positive, thus more negative. And so you actually would have reduced iron 3+ into iron 2+. So to be aware, cations can either be oxidized or reduced forms, because many metals can have multiple oxidation states. It's one of the cool properties of metals. It's partially what makes them really great conductors as well. It's that loose hold on electrons. All right, so dehydrogenase is a super common one. What do most of the dehydrogenases in metabolism do? Since we're talking metabolism in this page anyways, we won't for most of the class, but we will for now. What do dehydrogenases largely form in metabolism? You can actually do glycolysis or Krebs cycle, to be honest. Dehydrogenase. What are they going to be making? Maria's getting to the key point. If you want to, so the neat thing is, if you look at your entire process of glycolysis in the Krebs cycle, any time that you're forming NADH or FADH2, you have a dehydrogenase, right? Because the difference between NAD+ and NADH is you're moving hydrides, you're moving electrons. So all of those different groups in any reaction that's going to be making NADH, it's got to be a dehydrogenase. Exactly. So perfect. It's electron acceptor, you got it. In this case, it's that hydride that we're moving back and forth between different molecules. Cool. So that's a super common one as well. But of course, reductase, oxidase, those are easy roots. I do want you to remember dehydrogenase. That's a very common example in metabolism as well. All right, last one is transferase. Transferase, we can kind of course, use word roots here once again. You got it, transferase, right? So we're transferring. So we're transferring a functional group exactly as Ashley said. Michael's saying the same thing. Rose is saying you got it. So a couple different ones here. A lot of them are called transferase. Kinase, though, kinase is very specific and very prevalent. What is specifically kinase transferring? Excuse me. Phosphates. Once again, you got to know kinase is another very big part of metabolism, of course, causing phosphorylation or dephosphorylation, because, of course, if you're going to phosphorylate something, you got to pull it from somewhere. Often ATP is where we're pulling it from. So perfect. So no kinases for sure. Very, very common transferase. Or co-H transferase. Totally true, Stefan. Great point. Of course, moving your CoA groups, making acetyl-CoA. Or other CoAs as well. There's a bunch of them. All right. There we go. All right, so now the thing is, you don't need to memorize all the different enzymes that we discussed here. A couple key ones you do need to know, like kinases, no. Dehydrogenases, no. Lipase. In fact, what is lipase going to be chopping? Notice you can use word roots to figure out all of this stuff. Lipids, exactly, fats, right? So it's part of digestion, breaking down your fats. And so as long as you know your word roots and you know the basic categories here, you can pretty much figure out the function of any enzyme that you see on test day because it's a multiple choice test. Use that to your advantage. That's the coolest part of it all. All right, so let's jump into some questions. We're going to kind of move on and start really getting into the more specifics of inhibition and different, in terms of kinetics and all that. But let's do this. I'm actually going to open up a poll. I don't believe we have one on screen right now. Do you mind grabbing one of the other teachers and covering the download me in the lower portion of the screen? We'll bring up a poll here. We'll have you guys answer it. Once we've got it, we will discuss. So take about 30 seconds or so to solve this. Drop it on screen whenever you're ready to. Thanks for bringing that up. All right. Looks like we do have most people's answers in the polls. And as you can see, we've got about 90% of you, 91% of you saying answer choice A. Nice work. Of course, enzymes, remember, we said they're generally made of proteins. This is why I was referencing before that at this point in evolutionary history, they're largely proteins. But originally, they were all RNA. There's still a couple RNA enzymes around, but pretty much at this point, they are all proteins, at least the ones we talk about. But they were RNA in the past, and there's still a couple floating. We also know, of course, that many do require cofactors or coenzymes. We also know that they function at very specific pHs and temperatures, which is why, of course, they can be affected by genetic mutations. And many different genetic mutations that we talk about are largely due to basically mutating enzymes. Let's give an example. So how about the enzyme pepsin? Pepsin's a great example of an enzyme to talk about. So in pepsin, what would its primary pH and temperature be? Super low pH, you got it, because it works in the stomach. Now, why in the world would you want a protein to only really function at a pH of 2? Why not let pepsin function at any pH? What's the point of that? Why would we do that? Yeah, I like it. That's a good thing. Harpreet's saying it. Ariel's saying the same thing. Because if it got out of your stomach, which can happen if you get a gut wound, right? If you get shot in the stomach, all this chemicals start leaking out in your bloodstream. And if pepsin was active at normal pH, that means once it gets into your blood, it's going to start eating you. It start breaking you down. This way, and it's also the same in a very specific type of membrane-bound organelle. What membrane-bound organelle would you also want to control very carefully to not accidentally digest the rest of your cell? Yeah, you got your, lysosomes, right? Lysozymes would be the enzymes working there, but exactly true, because they also cause digestion. So you'll notice a lot of these different encapsulated things, even your stomach is a good example of a macro example. You want to control the pH so enzymes are only active in those one specific environment so they don't accidentally digest the rest of your body. Cool. Nice work, everybody. Cool. All right, so that's the first one. Let's do another question here. Clearing this out. Put your answers in the poll as soon as you're ready. All right, we're a little split here. I like it. We're pretty consistent here. A lot of folks are knocking out a couple key answer choices. More specifically, we're knocking out answer A. You're right. It does not change the delta G. We talked about that, which means it can't increase the forward or reverse reaction rate more. It's changing them both equally. So I mean, we're largely down to either C or D, but as you can see, we do primarily have people saying answer choice D, and I do completely agree with you. They do not react chemically during the course of the reaction. What's another way to say that? What's another way to rephrase D into what we talked about before? Yeah, they're not consumed, right? They're not used up. They're not broken down. They're unaffected. If they reacted chemically, that would mean you're changing covalent bonds primarily, and you're not changing them. You're not breaking them down. We don't change the thermodynamics. And they can be molecular weight as low as 1, because remember, we said actually a fairly common catalyst is protons. And protons, of course, are the only thing that have molecular weights of 1. So from protons all the way up to macromolecules and proteins, as we said before. Cool. So, oh, actually, I switched it around. It's not D. Actually, I said everything correctly, other than I switched the answer there. But remember, we said they do not react chemically during the course of the reaction. We said this. Of course, that's because they're not used up. They are regenerated, they're reproduced, or recreated. They're just not used up. But you guys got it. It actually is C. Everything I said, I just swapped the numbers there, but you guys are totally spot on. All right, I'll keep it going. But it is C there. It is C there. Remember, even protons work. Even protons work. All right, that breaks down the very first learning goal. The next two we're going to squish together kind of into the same thing. We'll talk a lot about the graphs and the properties. We'll get into the kinetics of enzymes for the last part of class here. All right, so when we talk about the kinetics of an enzyme, here, actually this is a little funny image. So here's a bunch of different soccer players. Of course, they all have different soccer balls, and they're going to, of course, kick them into the goal here is the net goal here. So now when we talk about the Vmax, what it's really referring to, what would the reaction of this really be? What would your reaction be here? That the soccer players are the enzymes catalyzing. We like to use metaphors. I don't know if you guys have realized this yet, but we're big fans of metaphors. How many goals exactly? So the reaction is getting the ball, which is the substrate, into the goal, right? And so now notice when we look at this, notice right now the only rate that we'll have of the reaction, of course, if you only have 4 soccer balls, these 4 different people are going to be shooting goals, and of course they're going to be getting points right there. And so now notice as you add more soccer balls to the pitch, notice as you add more substrate to the game, you can increase the rate of scores because now you've got like a dozen soccer balls and you've got one very pissed off goalie, of course, if you've got dozens of people with dozens of soccer balls all firing away at them at the same time. So then what happens once you start to add more soccer balls to the pitch? It's saturated. It doesn't make any difference because you already have every single enzyme, which is a soccer player here, has the only ball that they can have before they kick it. You can't kick 5 balls at the same time to get it into the goal. So you would call this a saturated system, which the way that we describe it is by hitting Vmax. It's the maximum velocity that the enzymes are taking. So it doesn't matter how much more you add. So of course, it's not going to change the rate of reaction. Notice there's loads, 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 loads. Doesn't make any difference. You're still going to be scoring as many as you have people. I agree, Rose. I like metaphors. I'm a big, big, big fan. I've made up a lot of different enzyme metaphors. We won't get into them today. It would take too long, but we could go for days with enzyme metaphors. I like the soccer ball one myself, too. All right, so that's kind of the main idea there. Notice Vmax, of course. That'd be enzymes are saturated. You've got as much substrate as possible, so adding any more isn't going to make a difference. All right. So that's what Vmax means. It's the maximum rate of turnover. It's the maximum rate of reaction, we can basically say. So now if we're going to actually graph this, so notice your y-axis is the rate of the reaction, your x-axis is the substrate concentration. What would this look like? Nice. Asymptote. Totally true, William. You got it. So notice it basically is going to top off right here. And so where it levels off, Mary, perfect, that's the Vmax. You got it. So right there where it actually levels off, that's where every single soccer player already has a ball. And so by adding more soccer balls, in this case, remember, soccer balls are the substrate, you're not going to increase the rate of goals being scored at all. You got it. So tapers off of Vmax, and that's what that is right there. So now, that's an important thing right there. Here's your Michaelis-Menten equation. You don't necessarily need to memorize this, but you do need to be comfortable working with it. Yes, I'm sorry, but there's algebra on the MCAT. Most commonly, we have to do algebra in the context of unit analysis and like figure out which ones are going to be the same units or not the same units. But you don't even really memorize it. It's mostly the idea behind it, and so we're going to get really into KM and what the heck KM means. So I think you guys know this, but what does KM actually mean? You could say it as a limit, Rosa. There's a couple ways you could describe it. I don't know if other teachers have one. I would call it actually a limit, personally. I like to use the calc terms to describe it. But you got it. It's the concentration of substrate at which you get half the maximum velocity. So I'm going to say it again. The Km is the concentration of substrate at which you get the half max velocity. So what you could then do to solve for this, notice if you take Vmax divided by 2, thus half the maximum rate, and then we do a little bit of algebra here. So notice what the first step that we did here was to first multiply both sides by 2, multiply that side by 2, cancels out your 2's, and then we're left with the 2 on that side of the equation. After that, we divided both sides by your Km plus substrate, of course, to move it to the other side. Actually, looks like we multiply both sides by 2, and we also multiply both sides by Km plus S. It's really scribbly right there. So it causes the denominator on both sides to cancel out. Effectively, you move the 2 to the right-hand side of the equation, and you move the Km plus S to the left side of the equation. Doing a little bit more algebra here, then you can solve, of course, by getting rid of your like terms there to, of course, break it down to find that Km equals the concentration of the substrate at which you get half max velocity. Now remember, you don't need to do a bunch of algebra on test day generally speaking. You can almost always find shortcuts to avoid doing too much work, but this is just showing you, of course, how you derive Km to actually equal half the maximum rate of reaction. And yes, you do need to know the basics of course, your algebra. Notice you cancel out Vmax on both sides, get rid of that right there. Then doing a little bit more math, you can find out. Actually, cancel the substrates right there. It leaves you with the same thing. So it's just canceling. Actually, it's just canceling. That's all we did there. All right, so most of you know that Km is the concentration of substrate at which you get half the max velocity. So if we wanted to graph that right there, would Km be on the x-axis or the y-axis? So would Km be on the x-axis or the y-axis? It's the x-axis. That's the trick to remember. Remember, Km is describing the concentration of the substrate that you get half Vmax. So Km actually is down there on the x-axis. Nice work. Cool. Yeah, and so as Debbie says, you don't need to memorize the equation. You know this stuff. You'll be fine. Mostly be able to interpret it and use it to your advantage, which we will do in a second here. All right, so there's your Km right there. Be aware what it means. All right, so notice, of course, we talked about this already, but notice the rate of reaction is slowing down, slowing down, slowing down, until finally when we get to the very top right there, notice that any more that you add is not going to change the rate. So we discussed that in the very beginning of this page, so we don't need to discuss it now. All right, so Lime River Burke, on the other hand, cool, perfect. And actually, yeah, Stefan's got a great point there. Let's talk about, I was going to do that later on, but we might as well do it right now. That's a good point to bring up. So notice that means if you have a very, very, very low Km, what does that mean about the affinity? The way to describe it. With a very, very low Km, what does it mean about affinity? Super high affinity, right? Because that means at a super low concentration, you're already getting to half the maximum rate, right? Whereas if you have a Km that's way down there on the right-hand side, far down the x-axis, that means that it takes a super high concentration of your substrate to get to your half max rate. Perfect. Perfect. You got it. Exactly true. Exactly true. All right. So there we go. Yeah, I like the way you guys are analyzing. This is perfect. All right. So now Lime River Burke's the other way that we see this represented. It's the double reciprocal of your Michaelis-Menten equation. Once again, you don't need to derive this on test day. This is just here to show you basically what we're doing. But by double reciprocal, exactly, it means that we're taking the 1 over both sides of the equation. Thus, we have 1 over v. And then you just flip the equation on the other side. And the reason that we do this, and actually brings up the takeaway point, it's so we can make a linear graph, of course, of showing the relationships here. So we'll linearize, I like the way you eyes the word, on the relationship between these two. So we don't need to go through the algebra. I'm going to show all the algebra right there, but to kind of like show the idea here. If you want, actually, do you guys know how to screenshot? In case you are weak at algebra, this might be a great example to practice doing this. You guys all comfortable? If you're on a Mac, you just hit Command-Shift-4. It'll give you a little crosshair. You can click and drag and take a picture of anything that you want on the screen. If you're a PC user, there's a print screen button on the upper right-hand corner. But so if you do struggle with algebra, this is a great example to try to derive Lime-Weaver-Burke from this equation. Generally speaking, you won't have to. You'll just know this stuff for test day. And so notice, generally speaking, when we have a linear formula, and this is what Ashley brought up, what is the standard form of any linear graph? What's the formula for any linear graph? Perfect. You guys know it. So it's y equals mx plus b, right? So notice y, of course, is referring to the y-axis, right? Whatever the y-axis unit is. The x is referring to whatever the x-axis unit is. M refers to the slope, And of course, B refers to the y-intercept. So notice that means the y-intercept of this double reciprocal is the inverse of V max. Thus, if you have, of course, a very low number there for your y-intercept, it means it's very high. If you have a very high number, it means it's very low because it's the reciprocal, right? It's the inverse. It's 1 over whatever it is. So then, what would your slope be here then? What would your slope be here then? What would slope refer to? Weird. So I guess we need to see what the axis is. So what's the x-axis? What's your x-axis if we do the double reciprocal? It's 1 over concentration of substrate. Exactly true. So notice, of course, there we go. We had it, someone said it first. Maria said it first. Nice work. Your slope then would be Km over V max. And we'll show you how you can actually derive the x-intercept and use this to our advantage. But you got it. So you got to know y equals mx plus b. That's a super common thing that we do see on test day. There's lots of different examples of linear graphs. So the way that we'd calculate the x-intercept, we already got the y-intercept, right? Y-intercept, right, that's where it intercepts the y-axis. It's the inverse of Vmax. The way you would solve for, of course, the x-intercept is all you got to do is plug in, of course, 0 for y, right? Because when y drops all the way down to 0 is when it hits the x-axis. So since we know the y-axis is 1 over v, just say 1 over v equals 0. Once you do that, do a little bit of math here, do a little deriving, you will find out that, of course, the x-axis there, let me do it all the way, there we go, is equal to 1 over, or negative 1 over km. Once again, I don't want to go through the algebra. We have a lot of concepts to talk about, so I'm going to focus on the ideas, not the math. The math is all available. If you want to screenshot it, go for it. But remember, you're not going to actually do this on test day. We want you to know what the graph means when you look at it. This is just here for complete sake, I guess you could say. All right, so notice the x-intercept refers to the km. Notice it's the inverse, negative inverse of km. The y-intercept refers to the Vmax. That's the take home there. Oh, perfect. Nice. Thanks for getting the 2W. All right, so here's a little graph right here. So notice if we want to actually apply this, the Vmax shows, of course, the y-intercept right there. But now remember, the y-intercept there is showing 1 over Vmax. So using the units here then, what would the actual Vmax be? So notice it's 1 over Vmax. So if you know if 1 over Vmax is equal to 0.002, so you got it, so that means Vmax equals 1 divided by 0.002, and 1 divided by 0.002 exactly is equal to 50. Nice work. Let's do a little bit of math fun here. So if you ever struggle with formulas like this and you want to figure out a way to actually derive it quickly, think of it as percents, right? This is 2%. What do you multiply 2% by to get to 100%, thus 1? You'd multiply the bottom by 2%. What do you multiply 2% by to get to 100%? Just 2%, right? You multiply it by 50. So notice if you multiply the bottom by 50, multiply the top by 50, that of course brings you right back to having 50 on top because we just have 50 times 1, and the bottom turns you into, of course, 1 or 100%, which is equal to 1. Perfect, exactly. Debbie's got the recap for you right there. But you guys all did it in the chat, so you're perfectly fine. Aha, totally. Makes it way easier. You've got to have ways to kind of get around the math. Because it is a pain in the butt. The other way you can do it is turn it into a fraction. 0.02 is equal to 1 over 50, right? It's 1/50th. And then, of course, you could just flip the reciprocal of that to get it into 50 as well. Loads of ways you can do math. Perfect. Glad to hear it. Oh, man. Percents make a lot of math a lot easier to think about. I'm a big fan of using percents whenever it comes to decimal math. Makes your life easier. Cool. And then we do the exact same thing for km. Actually, let's try it out. So then what is your km then? If negative 1 over km equals negative 0.5, bingo. That means we have, perfect, 2. Notice the negatives cancel out, right? So the negatives will cancel, and you'll be left with 1 divided by 1/2, and 1 divided by 1/2 means the inverse of 1/2. And of course, so it flips it up to 2. You guys got it. Good work, team. All right. So now, notice here, let's just talk concepts. We won't do the math anymore because I do want to focus on ideas here. But so notice, if we decrease the y-intercept, what does that mean about the actual reactivity here? Has it gone up or has it gone down? We decrease the y-intercept in this new graph. thus the reactivity has gone up. Remember that the y-intercept is the inverse of Vmax, so by decreasing the y-intercept, you've increased the velocity of this reaction. It's gone faster, exactly. How about here? So if your Km goes down, notice it's getting more negative. What does that actually mean about the reactivity then? It means it's a higher Km as well. And if you have a higher Km, what does that mean about affinity? Lower affinity. Perfect. You guys know your stuff. Rock stars. All right, let's do this question. This will kind of answer everything for you. Let's do it. All right, it's actually most of us already answered. Good work. So notice we get a bunch of data here, and all we're really doing is it says that we're solving for Km and Vmax respectively, right? So we got Km there, we've got Vmax right there. Now if you flip through it, notice Vmax looks to be right about 30. Watch your units there. Notice that it's nanomoles per second, right? That's going to make a very big difference because what did you notice about your units and all the answer choices? Notice this is nanometers, this is nanomoles. The answers are all micro. That might make a world of difference for your answer. Be very careful. And they do that a lot on test day as well. All right, so that means, of course, if that's your Vmax, that means that the Km is going to be, or the half max velocity is right at 15. So we drew a little line there. It looks to be a pretty darn tiny number. So Km's all the way down there. Now, notice the reason that we had most people selecting A and not B, notice what the difference is between A and B. It's a difference of a factor of 10 to the third power. If you said A, you're totally right if it was in nanometer. Nanomolar. Capital N means molar, right? But exactly in this case, though, it's in micro, so whatever your answer is, you need to divide it by 1,000, as Ashley says. And so everyone that said A, you were in the totally right game. You had the right idea. It's just, of course, that you got to convert your units. You got to be careful. Super be careful with those units. But we were pretty torn between A and B there. Nice work. All right. That brings us to our final learning goal of the day. Let's talk about inhibitors. So we've got 4 things to talk about for the last part of class. We'll talk about the 4 different types of inhibition, and we'll just review showing what that actually looks like on a Lineweaver-Burke. This is some high-yield stuff. This does show up quite a bit. All right. So here is an example of a passage. We're not going to spend too much time really going through the whole passage. Instead, I'm just going to kind of give you the outline. We don't really need it. It's fine. So we're talking about these organophosphates. So history of them. The short thing is organophosphates, good thing, bad thing. Bad thing. Super, super bad thing. Exactly. So they're toxic. They cause major problems. They're actually in pesticides. You'll notice them actually working in, if you do work in a rural area, you might actually have patients come in that do have organophosphate poisoning, and you'll have to deal with that. We can talk about it pretty soon. But the main thing is, it looks like, what do they really kind of act on? You just kind of scan through it, look for the sciencey words. It looks like they notice a trend, and they theorize. That's the kind of strong language that we look for here. They, of course, theorize that it is all about effects through acetylcholine esterase. So if they're going to, of course, inhibit acetylcholine esterase, what would we end up with too much of? Too much acetylcholine, right? Acetylcholine esterase breaks down acetylcholine, so it sounds like the main idea here is that we're going to get too much acetylcholine. All right, so that's the main idea there. So we have an experiment here. We're going to use this Beer's law. If we ever need to use it, we can come back to it. As I said, we're going to focus on the content here, not on the passage outlining. We do that enough in class. And of course, then we get the results in the very bottom. So remember, anytime you get an experiment, what matters most is the results, right? So here we go. We got substrate. We're comparing it to velocity. This is the kind of stuff we care about, and this is the kind of stuff that we're actually going to talk about the most in terms of the questions. So we've got Beer's law, we got an experiment, and we're going to figure out the velocity of this reaction, of course, how we can see how it affects various things. All right, so once again, we're not going to worry about outlining today. We're going to focus on the content. All right, so here's our data right there. Here's the awesome thing. So now I want you guys just to tell me, using your knowledge that we just talked about, what's being changed here? So what is being changed here? Nice, Vmax, exactly. And with this inhibitor, what's happening to your Vmax? With the inhibitor, you got it. The Vmax is going down. It's decreasing. So note, remember, this is a Lineweaver-Burke graph, right? So of course, it's the inverse of everything that we talked about. So the way that we know it's the inverse, if you weren't sure, notice that the units are weird because it's seconds over molarity, right? Generally, a velocity is a molarity per second. And so since we switched the units there, you know that it is the inverse there. You guys got it. All right. So as you can see, the Vmax went down. Add inhibitor, Vmax went down, Km stayed the exact same. That's what this is really all about. All right. So in order to solve this, we got to talk a little bit about inhibitors. This is going to be the main last part of class. We'll go through a bunch of different examples of inhibition, then we'll come back and actually solve question number 5. So let's actually solve this. So here's the last thing we're going to talk about. As we said, it's all about inhibitors. So first is competitive, and once again, we covered this at the beginning. Competitive inhibitors compete for what? Competitive inhibitors bind at the active site. Perfect. So competitive inhibitors bind to the active site, or they compete for the active site and prevent the substrate from binding there. So you can overcome competitive inhibitors by basically flooding them out. If you add a crap ton of substrate, then all of your enzymes are going to be fully saturated with soccer balls, right? And so it won't matter if you throw bowling balls onto the pitch. Yes, that's right. You could think of bowling balls as a competitive inhibitor to the soccer ball analogy that we said before. You cannot kick a bowling ball into a goal unless you're crazy strong, and I'm assuming no one's that strong. That's a good example. So of course, notice this increases the Km. But notice the Vmax does not change. So the Vmax doesn't change because it does, like, remember, if you add enough substrate, if you add enough soccer balls, the soccer players on the pitch are not going to even notice the bowling balls laying around. They'll be basically forced to just keep kicking soccer balls all day. There we go. And so this is what you need to remember. This is what you want to take home. Competitive inhibitors, they increase Km, thus, they increase Km, thus you need to add more substrate to overcome them. That's what Ashley said. That's your take home right there. So how about non-competitive inhibitors? Actually, we covered this one before too, right? Non-competitive inhibitors, I like it, they bind to the allosteric site, right, which causes a conformational change. There we go, Tadell's got it exactly right. It decreases your Vmax. If an inhibitor binds to your allosteric site, your enzyme's not going to work as well. It can't run as fast as it wanted to run. It's like if you broke a bunch of the soccer players' legs now, all of a sudden they're not going to be able to kick as many goals as they were kicking before. So it doesn't matter how many soccer balls you throw on the pitch. If their legs are broken, they ain't going to be able to do a thing. They're going to be weakly kicking with their non-dominant leg. And of course, the concentration will go down. So it will decrease the Vmax, as you said. It doesn't matter how many soccer balls you add, ain't going to make a difference. To stick with the same metaphor we've been using the whole class. All right. So those are the two biggest. Those are your highest yield. Those are the most important for test day right up across the board. All right. So now the more rare ones, you won't as likely see these on test day. But the ones that you can also see is mixed inhibition. Yeah, and actually, notice as Ashley said, pointed out there, Km is unchanged, right? Because there are going to be some soccer players still on the field that are still going to be kicking balls, and they're still going to be perfectly fine, right? Their affinity is no different. You only change the Vmax because some of your enzymes are having broken legs, and they're not able to do the thing that they're supposed to do. Let's stick with the metaphor. All right, so that's your take-home. Screenshot, important. Ashley's got the take-home there. Decrease Vmax. Of course, don't change Km. All right, so now mixed, as you would expect, mixed is just super messy. So mixed, the one thing you do need to know, what is the one consistent thing that changes with mixed? So it actually binds to the allosteric site, causes conformational shift, actually binds with same affinity to either the to the enzyme or the enzyme substrate complex. There we go, Ashley's got it. Either enzyme or enzyme substrate complex, exact same. And your KM sloppy. KM might change, might not change. The one thing that will definitely change, though, is your Vmax. So the thing to be aware with mixed inhibitors, because they're a little bit wonky, you're going to, no matter what, decrease Vmax. So you will definitely decrease Vmax. We don't really know about Km. You could either increase or you could decrease Km, and since it's sloppy, it's less likely to show up on test day. If the inhibitor prefers the enzyme, you're going to increase the Km. If it prefers the enzyme substrate complex, you actually decrease the Km. All right, so these are less common. If you don't geek out on these if you don't want to, but the one thing you definitely need to know is that they are definitely going to decrease Vmax just like non-competitive inhibitors did. All right, so the last type of inhibition, do you guys know the last type of inhibition that we're going to talk about today? Final part of inhibition. You got it, uncompetitive inhibition. Nice work. So uncompetitive inhibition actually only binds to the allosteric site of the enzyme substrate complex. And it's going to, there we go, exactly true. It's going to mess up both your Vmax as well as your Km. Nice work. And so uncompetitive is a little bit different. You got it. Basically locks it in, Mary. That's a great way to say it. So once they're bound together, your inhibitor binds. They are basically stuck that way. So it prevents the release of the substrate. Thus, it decreases Km and Vmax. And so you guys already know the answer to the question. From the graph that we saw before, what kind of inhibitor is this? What type of inhibition is this? The last question of the day. You got it. It's non-competitive. Notice that the Vmax is going down, but the Km is unchanged, which means you guys are spot on correct. It is non-competitive, and it's because of that Vmax. Nailed it. All right. Good work, everyone. That is the highest yield, most important stuff that you need to know for test day. We're going to take a short break. We'll come back and jump into cars. If you have any questions, hit us up in the meantime. We're going to be running the Q&A. Nice work, team.